Okay, smear preparation and staining. Now, some of this is going to be a repeat of the staining I, that I just did for you guys, but I'm going to go into more detail with the gram stain, and I'll go into further gram detail a little bit later on because these are important stains, and I want you to get a basic understanding. To give you a basic understanding, then we're going to go into a little more detail. Okay, so bear with me on this. I want you guys to get this. So when you make a smear, we want to make sure that it's thin. We're three goals. We're making sure that the cells are going to adhere to the slide. Heat fix it, as I said. You don't want to heat it too much. You don't want to shrink these cells. You don't want to distort them. You want to be able to air dry them before you basically let the slide air dry before you heat fix it so there's no distortion. And you don't want a thick smear because you can't see. Okay, and there are some YouTube videos, that I want you to look at. All right, so what we can use solid media. I showed you that plate with the colony. You can add a drop of water to the slide and then use your inoculating needle to go in and pick up one colony, just a little touch of it, want a lot on there, and then mix that bacteria with the water and spread it out as thin as possible, and then you let it air dry, and then you heat fix it and you can stain it. Liquid in media is much easier to work with in terms of you've got a liquid, so you don't need an additional liquid. So you go in and use a sterile loop, get a couple of loopfuls of the broth onto the slide, and then spread it around again. Let it air dry and heat fix it. And make sure you label your slides, because here's the thing. You need to label your slides so you know what the smear is, number one. Got it? But I always label it because some of the slides have a little frosted edge so that you know which is top of the slide or the bottom. But if it's just a plain slide, you need to mark on it. I always tell my students, we get a wax pencil and we mark the bottom of the slide, because when you put the slide on it, when you actually air dry it and before you stain it, you can't see where you've put the material. And you're like, okay, which side of the slide is up? And if you can literally stain just your wax pencil or whatever you're using. So always mark the bottom of your slide, kind of make a little oval on the bottom of your slide so that you know this is where I'm going to put the specimen so I can find it, once I get it stained, because it may not be obvious. If you've made a really good thin smear, it may not be obvious that's where it's at. So again, we have simple stains, we have differential stains, we have structural stains, and we've gone through this. Simple stain, one color, differential, at least two colors, usually two, can be more, usually two, to differentiate between two or more organisms. And then we can do a structural stain like a Flagella or endospore stain. And they work because they contain chemicals that impart color. And again, as I mentioned, the bacterial cell has a slight negative charge. And so we can use positively charged stains, or what we call basic dyes, that will bind to that cell, that be attracted. Or we can do a negative stain, which is an acidic stain, which is negatively charged, and it will repel, like when I talked about the last slide, the capsular stain, right? So we can do it either way. When we look at a simple stain, again, one color, size, and shape. That's it. These are pretty good size bacilli. They're not tiny. When you do a simple stain, again, I mentioned several different colors. This is bacilli stained with crystal violet. It's a very deep purple color. And this is the color that we use in the gram stain. Just FYI, we'll get into. We can use safranin, which is a red color. This is also used in the gram stain. And you can see that nice red color. We can use carbofusion, which is a fuchsia color. It's used in the acid-fast stain that we'll talk about in a few minutes. We can use methylene blue, which is a very light blue. But again, in all these different simple stains, size and shape, that's all we can tell, okay? We get into a differential stain, we have more than one color with a decolorization step in between. So we're using multiple dyes to distinguish between the different types of organisms or cellular structures, whatever they may be. So I'm looking here and I have gram-positive cocci and those lovely gram-negative bacilli again, okay? But you can see the bacilli, you can see the diplococci here, and one singly, and there's your little cluster, all right? So the most common differential stains, again, gram stain, acid-fast stain, endospore stain, although that is a structural stain too, okay? So gram stain, let's get a little more in depth on this. I want to explain to you how this goes, and then I've got a whole nother thing on gram stains specifically, but I want to introduce you to the gram stain. So developed by Hans Christian Gram. And again, this is the most critical differential stain we use. It's quick, it's fast. We can do it in just a few minutes and give a doctor. I mean, this is a stat stain that can be ordered. Most things in the microbiology lab are not stat. You can't order a stat culture because it takes 24 hours to grow. But you can provide us with something, a swab or something, and ask us to do a stat stain. Okay? So this, again, it's going to divide these organisms into gram-positives and gram-negatives. And we've already talked about the gram-positive have that thick peptidoglycan layer. The gram-negatives have that thin peptidoglycan layer, and that LPS, that outer LPS, that lipid polysaccharide membrane. All right, and we're going to wash that out. We'll talk about it. It's critical in treating bacterial infections. I mean, again, it can be reported to the provider on a stat basis, unlike a culture. All right. What it gives them is the gram stain reaction and the shape. So if you get a report, again, I talked about this, gram-positive cocci clusters, you're looking at a staphylococci or something. Gram-positive cocci chains, very suggestive of streptococci. Gram-negative bacilli, you got a whole bunch of them. But it puts you in that this is a gram-negative, and we would treat this with a different antibiotic. Whatever is seen as reported, whatever we may see, bacilli are not reported in chains because they only divide, remember, in one plane, and they cannot form clusters. So we do not report them bacilli in chains because they're going to be in chains. So we don't report them as that. All right, so let's look at this actual gram procedure and what's happening. And again, I'm introducing this to you. This is the first time. I like to do this in pieces and parts. And then, next week, we're going to have specific labs on the gram stain and the acid fast. But I want to introduce this to you and start with that. So when we work with gram stain, as I mentioned, we have crystal violet and we have safranin as the particular dyes. And you can see what happens. So you take the smear and you will flood it with crystal violet. You'll prepare the slides, you'll let it air dry, you'll heat fix it, and then it's ready to go. And you flood it with crystal violet. And that will, everything on the slide will pick up, whatever it may be, will pick up this purple dye, okay? And then you will wash this off. You'll let it sit for a minute and wash it off. And what the crystal violet does is, again, this is a positively charged stain. It's going to be attracted to our basically negatively charged bacteria. So it's going to pull this dye into the cell wall of this bacteria. Now, what we're going to do then, after we've washed it, is we're going to add iodine. This is a mordant. Think of a mortar, in between bricks, okay? What this is going to do is make an insoluble complex with the dye and make it so that it is less soluble. Kind of sticks it in there and makes sure that it's going to hold. And at that point in time, these organisms are still all purple because you're purple and you're going to put iodine on it and it ain't going to pick up the iodine color. All right, wash that off. Then, and we're going to go into specific times and everything like that again in the next video I'm doing. We're going to decolorize. This is the decolorize step. And what this does is it washes away the stain. And this is going to be the piece that differentiates the gram-positive from the gram-negatives. You very gently, very shortly, short time, wash this slide with alcohol. And what happens is that in 10, you can see this 10, I don't even do 10 seconds when I cook. There's no way I'm doing 10 seconds on a gram stain. No. 1 to 2 is enough. What this does is it basically washes away that LPS layer from those gram negatives. Just dissolves them. But your peptidoglycan layer on your gram positive is so thick that there's still some left. You haven't washed out all that crystal violet. Now, if you leave this alcohol on too long, you will go through that entire peptidoglycan layer. Just FYI, you can. But that's why we don't put it on there for too long. We're just going to let it hit it with it and wash it off. And basically that LPS will dissolve. And at that point in time, your gram positives will still be purple, but your gram negatives will be colorless because all the dye has been washed out. Then you counter stain. Safranin is your counter stain. We're going to counter stain with a color that is on the opposite end of the color wheel. Talk about that in the gram stain too. There's no use counter staining a purple with a blue. It's hard to see. So you're going to counter stain with something that's obvious. So we're going to use safranin, which is red. So we got a violet and a red. And what will happen is the gram positive cocci is still all full with the crystal violet. So it's not going to pick up the safranin. But you've got that whole second layer on the gram negatives that will now pick up that safranin. And that's what makes them look red. Okay? And that's how you see the difference. And again, I'm going to go into this in greater detail in the next one. So this is how you get this lovely one, the same slide that you've been seeing and seeing. But you can see the difference now between the gram positives and the gram negatives. And now you understand why they're staining the way they're staining. Okay. Acid fast. Again, I've got another whole thing with acid fast. This is an introduction. Okay. Acid fast is used to identify bacteria that resist decolorization with acid. Okay. Acid fast means the color held fast even though we used acid to decolorize. That's how they came up with the name for this acid fast stain. Okay. It is primarily used for Mycobacterium tuberculosis, which causes tuberculosis, and Mycobacterium leprae, which causes leprosy. So most 99% of the time, what we get an acid-fast stain in it is on a sputum. And we're looking for these acid-fast organisms. Hopefully we don't have tuberculosis. They're reported as either bacilli-seen, acid-fast bacilli-seen, or no acid-fast bacilli-seen. That's how it's reported. Okay? And again, it's called acid-fast because the color, which we're going to use this carbofusion, stays in even though you've used acid alcohol to decolorize. You've used an alcohol with acid in it. Okay, so here's the procedure. You put it on for 30 seconds. You let it sit there again. Don't necessarily have to heat it, but yeah, there's a heating. Basically, there's some cold ones, but you have a beaker of water that's boiling and it's set over this beaker of water, boiling water, steaming, and it opens it up a little more. And you decolorize. Once that is done, you wash that off and you decolorize with acid alcohol for about 15-20 seconds. You do let it go, okay? And then you apply the counter stain, which is the saffron, the methylene blue, which is this nice blue. And what we're looking for is these bacilli, these pink bacilli. So here we go. You can see the methylene blue, that blue background. There's your acid-fast bacilli. So this would be reported as acid-fast bacilli. Again, I've got another whole lecture early on this one. And then the structural stains, we kind of already went with this, with the endospore stain. Again, Bacillus, Clostridium. It helps us identify some of these guys, Bacillus anthracis, which is cause anthrax. We've had anthrax scares there a while ago. Clostridium tetani, Clostridium botulinum, cause of tetanus and botulism. So there are times when we really want to go in and look at this stuff. We'll talk about that a little bit more. But endospore stain is we apply the primary stain, which is malachite green, and we apply heat to let it get in there, and then we decolorize with water, and then we add saffron, which will be picked up. And you can see with the endospore stain will pick up that lovely green. All right, and then capsular stains I've already mentioned a little bit. That we talked about the thick capsules. Capsules are water soluble and repel most stains. So we use a negative stain. We use, as I mentioned, India ink. And basically it will cause a halo around these organisms. It's ordered as an India ink prep on spinal fluid. We're looking for Cryptococcus neoformans, which causes meningitis, cryptococcal meningitis, and that's India ink prep's always ordered every spinal fluid tap that we get. Well, all I can say. And then this is what it looks like. And you can see this dark ink is going to be darker than that, but you can see these yeast cells, okay, reported as positive for encapsulated yeast cells, and that tells us that it is a Cryptococcus. And again, the flagellar stains, we mentioned that the flagella, they're very thin. They need a special stain. We can use a mordant like tannic acid to thicken them. They're used in gastroenterology to help identify Helicobacter pylori. Okay, from gastric biopsies. A gastroenterologist may do this in their office. Helicobacter pylori causes ulcers. Okay, these are the organisms that cause it. So they're typically reported out with the description of the flagella, just FYI. And we've already gone through this once, and so you need to learn these, okay? And then you can see Helicobacter pylori with the flagellar stain, okay? And you can see it, all right? And that's it for this. Again, just a reinforcement of what I've been telling you. I want to make sure that you get this down. You get bored with it, I'm sorry, but it's important. You're not in a lab. So I want to make sure you get this.